Thank you very much for the introduction. Again, thank you for inviting me. It has been a great day. My highlight was definitely Maria de la Bora's talk. It was very inspiring, and I will dedicate the first baby I treat with stem cells to her. I would also like to thank the translators. You made a great task. I feel I could give this talk in Greek now. And I would also like to thank Christian Speer because he provided the perfect introduction to my talk. I can shave off 10 to 15 minutes of my presentation. He showed very nicely that BPD is a multifactorial disease, and he stated very nicely at the very end that it needs a multimodal approach. And this is what the cell-based therapies are all about. So I will skip through the first slides to save time and offer more time for discussion. Because you have heard all this and seen even some of the same slides. See that? 10 minutes. Thanks, Christian. And so I will go straight into the cells that we are so interested in because it appears that they have this pleiotropic effect, allowing a multimodal approach for this multifactorial disease that is BPD. I introduced them briefly in my first talk. These are these mesenchymal stromal cells that are niche cells for the hematopoietic stem cell, but in the late 90s have been found to have repair properties. And these discovery of these repair properties of these mesenchymal stromal cells also offered alternate hypotheses regarding repair mechanisms in the lung. We knew from the 70s that in the distal lung there's this alveolar type 2 cell that produces surfactant, but that is also able, upon injury, to proliferate, de-differentiate, and become type 1 or type 2 cells, and thereby restore the homeostasis of the alveolar interface. With the discovery of these mesenchymal stromal cells, it was hypothesized that upon injury, bone marrow-derived cells could sense danger signals from the lung, migrate to the lung, and replace the dead cell and thereby restore homeostasis in the distal lung. And so many investigators have used this same animal model in mice and rats that are exposed during the neonatal period to various levels of oxygen. And you can see how this causes a dramatic disruption of the alveolar growth and lung vascular growth as well. And these models are imperfect, but they allow for rapid proof of concept establishment. And this is what investigators around the world did. They used this model to test either a prophylactic or a rescue approach with mesenchymal stromal cells. Here, these are the two Princeps papers, one in which bone marrow-derived MSCs from rats were given to neonatal rats exposed to oxygen. You can see here very nicely the dramatic arrest in alveolar development and prevention with mesenchymal stromal cells. Likewise, dramatic arrest in lung vascular growth and partial prevention with mesenchymal stromal cells given intratracheally, similar to surfactant. The group in Boston, headed by Stella Kourembanas, you will like this last name, she is Greek, she gave intravenous mesenchymal stromal cells, showed the same therapeutic benefit on lung structure, but she also showed that there was a reduction in lung macrophage and lung neutrophil influx, suggesting that one mechanism of action of these mesenchymal stromal cells is anti-inflammatory. And you have seen just in the previous talk by Christian Speer how these neutrophil and macrophages contribute to the pathophysiology of BPD. But of course, a clinically relevant source of mesenchymal stromal cells is the umbilical cord or umbilical cord blood. We are more interested in the umbilical cord, the actual tissue, the Wharton jelly, because it yields much more mesenchymal stromal cells. and all the advantages of the source of MSCs is listed here. The most interesting one is that these mesenchymal stromal cells seem to have superior repair potential than you and my bone marrow, because we unfortunately have aged already. And it appears that the younger the cells, the more powerful the repair potential. And so we performed the same series of experiments, this time with human, look at this, with human cord-derived mesenchymal stromal cells. In this experiment, we used them as rescue strategy, where we inject cells intratracheally after established lung injury, and you can see rescue with MSCs or pericytras are precursors of mesenchymal stromal cells. Importantly, we let some of these animals live for over 6 months because we wanted to see if these cells were able to induce tumors. Here we performed whole body CT scans, since we have sliced all the tissues, and we could not find any tumor in these treated animals. We do exercise capacity at 6 months. This is the equivalent of 30 years in humans. You can see here very poor exercise capacity when we have these animals run on a treadmill, persistent benefit of mesenchymal stromal cells, and no adverse effect of the control animals that were treated with these stem cells. Likewise, for the lung architecture, you can see here at 6 months, still persistence of the arrest in alveolar development, consistent with what Christian Speer showed with the early aging hypothesis, or actually persistence of arrest in alveolar development without ever having any growth. And persistence of the therapeutic benefit of MSCs given in the neonatal period, and no alteration of the lung architecture in control animals, suggesting safety of these cells, at least in this animal model. Here I showed you the dramatic arrest in alveolar development. You may recognize this picture, and you have here some improvement that is still far from the control animal, but we know that the lung can compensate for 80% of loss of vasculature before having pulmonary hypertension, at least in the adults. So here, the small improvement in lung vasculature can make a big difference, in fact. This is work from the group in Seoul that has published the first phase 1 clinical trial. These were their precursor or preclinical experiments, where they also showed prevention of the lung injury compared to untreated animals with cells given either peritoneally or intratracheally. And they used cord blood-derived MSCs. And so here there was enough literature now that we performed a systematic review and meta-analysis of preclinical studies, not clinical, preclinical studies. And so this was work by a fellow in our NICU who screened through 1,000 data. He was able to retain 24 papers for analysis. And here I just show you the forest plot for effect of MSCs on lung structure. And you have listed here all the papers. And you can see here that except for 3 studies where the cells were given intranasally, mesenchymal stromal cells clearly improved lung architecture. And this is the most robust and transparent way of presenting the literature. But we will get back to this towards the end of my talk. One interesting aspect that was a bit scary for us when we looked for the first time was that we could not find the cells in the lung. Here I show you an untreated lung, and one lung that was treated with MSCs that we had labeled fluorescent with a green fluorescent dye before injection. And here we look at the lung 14 days after the administration, and you can see that we can see a little bit of green, suggesting that some cells were retained after 14 days. But when we do the careful analysis, we can see that very, very few cells are retained in the lung. So how do we reconcile this very striking therapeutic benefit with the little cell retention if we believe that this hypothesis is true, that cells home to the bone marrow, and then what they do, they self-renew and they differentiate. This is what I told you, what MSCs or what stem cells do. In fact, what happens is that these cells do move into the lung, but then they monitor the environment. They talk to the surrounding cells, and depending on the cues that they receive, they will dispense various factors that modulate then the repair response, suggesting that mesenchymal stromal cells do not engraft, but act probably via a pyrocrine effect. And this could be easily studied again in this animal model, where instead of culturing and then taking the cells and injecting them into the animals, we actually culture the cells and then take the cell-free supernatant or conditioned media, basically taking the juice of the cells and inject it into animal models. And we could see that similar to cell therapy, just taking the juice, here you can take the picture, just taking the juice would have exactly the same effect as the cells. Here I show you a model where we ventilated neonatal mice and we gave them also LPS, so we could mimic a little bit closer the inflammatory hyperoxia and ventilation-induced lung injury in these animals. And you can see the dramatic lung architectural alterations, no effect of the placebo, but a dramatic benefit of conditioned media from mesenchymal stromal cells giving to these animals just before the ventilation. And these are the statistical data. This is how my daughter sees it. She was 12 years old at that time and drew the front page of Pediatric Research. I told her what this was about, and this is what she came up with, that we just need to give those baby stem cell juice and we would repair the lung this way. And Stella will show us that this might actually be possible because she's the pioneer in exploiting extracellular vesicles that are present in the conditioned media. All cells produce extracellular vesicles, and there is one extracellular vesicle that are called exosomes, that are nano-sized derived membrane particles that every cell sheds. And this is how cells communicate with each other. Now, if you're a mesenchymal stromal cell and you're a repair cell, then maybe this could be a way how you mediate the repair effect. And so these exosomes contain various factors, but also microRNAs. So you can modify the host cell. Here we used repeated ultracentrifugation to gain these exosomes. This is how they look like on electron microscopy. And then if you check for their size, they are in the nanometer range, opposite to microparticles that are larger. And then they also express certain specific proteins such as TSG1. Here exosomes that microparticles do not express. And you can see right away here again, manufacturing will matter. These are the results in our ventilation-induced lung injury model. Exosome significantly improves the lung architecture, whereas exosome-free conditioned media or placebo has no effect. And here I show the data from Stella Coimbana's lab in the O2 model. Hyperoxia, arrest in alveolar development, and marked effect of Wharton jelly-derived MSC exosomes or bone marrow-derived exosomes, and no effect of a control cell exosome, human dermal fibroblasts, suggesting that the benefit is really due to these mesenchymal stromal cells. So this is the current hypothesis, that upon injury, MSCs get activated and then act like a medicinal MSC, or act like smart pharmacies, coming back to the analogy of smell phones. And you can see that these cells are strong immunomodulators, but they also have other effects, including improved wound healing. They also produce a variety of factors, VEGF, HGF, IGF-1, and we have heard today that at least IGF-1, for example, can improve or decrease the incidence of BPD. But here the cell doesn't release just IGF-1, it releases a host of healing factors. Again, coming back to the multi-modular approach of mesenchymal stromal cells. Immediately you can recognize that beyond BPD, there could be some other complications of prematurity that could benefit from mesenchymal stromal cells. I will show you two. One is brain injury, because BPD is an independent risk factor for adverse neurodevelopmental outcome. And so here we thought, okay, what happens to the brain stem cells in BPD? So Marissa took our O2 model, and instead of being interested in the lung, she actually isolated neuroprogenitor cells. And Marissa showed that in hyperoxia, the self-renewal capability of these neuroprogenitor cells was dramatically impaired. And then she does behavioral study and shows that after 6 months, these animals behave abnormally. And if she gives exosomes to the animal model, she can preserve function of these neuroprogenitor cells. So this will be very interesting to see in larger trials to see if we also have a benefit on neurodevelopmental outcome. But for this, we need a larger number of babies. And then we also looked at the effect of MSCs in a neonatal sepsis model induced by E. coli. And here, human umbilical cord MSCs were given 30 minutes after inducing the sepsis. And mesenchymal stromal cells improved survival dramatically, increased bacterial clearance, and probably they do so by secreting antimicrobial substances such as LL37. So I just showed you probably improved neurodevelopmental outcome and a very effective way of fighting sepsis. This is in stark contrast to dexamethasone that impedes neurodevelopment and increases the risk of sepsis. So, of course, the excitement was big in neonatology, and the group in Seoul did the first phase 1 trial where they assessed safety, feasibility of cord blood-derived MSCs, allogeneic, 2 doses, intratracheal administration, around 10 days of age. And they showed that this technique was feasible and there was no adverse events due to the cell administration. And I think they currently finished enrollment of their phase 2 study, and we eagerly await the data, maybe in April in Würzburg. Was that a good publicity? and also the same group did 18 month to 2 year follow-up, which is crucial for this new therapy, and showed no toxicity. Just this month, the group at Rush University in Chicago published their results of phase 1 study. Same cell product, exactly the same. They also showed that this administration intratracheally was feasible and that there was no toxicity. So I will just finish with a word of caution, because of course these preclinical data are very exciting, but here are the data actually about the increased failure of clinical translation of promising drugs. This first author, who I actually completely forgot that he's also of a Greek origin, actually showed how few promising drugs make it into the clinic and receive actually licensing. And the reasons for failure are listed here. And with this incubator concept, we try to mitigate the risk of translating cell therapies into the clinic. So I showed you this template, our roadmap to successful clinical translation of MSCs. And I feel much better now after Maria's talk, because I know that it requires a lot of patience, perseverance, and a little bit of craziness. But we go by it in a very rational way. And even though I'm very excited about this, I'm also very cautious, because I do believe that we have not figured out exactly what is a good repair cell compared to amongst all these cell products that we have. I showed you that systematic review and the dramatic effect in the forest plot of the therapeutic effect. This is the risk of bias analysis, which tells you a little bit of a more cautious story. Because here it shows that the risk of bias is often unclear in those preclinical studies, suggesting lack of rigor in our bench side projects with poor preclinical study design and lack of transparent reporting. And ARRIVE guidelines have now been proposed to allow for more rigorous studies at the bench, and this is what we're doing now. The other interesting aspect was that we looked at all these 25 studies. 25 studies, they were all in the same animal model. So here we have the luxury and the opportunity to work with the only baboon model that exists on the planet. And this is thanks to Dr. Steve Seidner, who approached me one day. And when he told me that he was in San Antonio, I knew right away. And I said yes to whatever you have to propose. Because this is really as you can see from these few pictures here, a very clinically relevant model that allows you to gain all these information that you would actually gain in a clinical trial: feasibility, safety, and eventually also efficacy to obtain regulatory approval. So these baboons are delivered prematurely, they are intubated, they receive catheters and then surfactant, and then they are ventilated. You can see here, this looks almost like a human, except for this little appendix here that we don't have. And then these animals are ventilated for 2 weeks. And then we also can take analyze the lung tissue. So in these animals, so here we gave the cells intravenously. There was no infusional toxicity except for the first one, where cells were given intravenously directly like this. Of course, you have to do it slow, and you have to do it with a automatic pump to avoid clumping in the lungs, like pulmonary emboli. And then we can look at the further safety efficacy over week 1 and 2, and then look at tissue analysis. And here you can see how heterogeneous the lung injury is in this animal model compared to the neonatal rat or mouse. And this resembles much more the complex lung injury picture that Christian Speer also showed at the beginning of his talk in the autopsy specimen. And here we will do careful lung stereology, which you can only do in Germany. And I hope that we will soon have the data of the lung architecture to see whether there's a therapeutic benefit in this animal model. What I can also show you is that these cells definitely do not engraft. They don't stay in the lung. Here we used forensic methods to see if there was any material related to the MSCs in the lung, and we couldn't find it. Again, cell manufacturing is crucial. This slide is crucial because it shows how much work still needs to be done, because the secret behind a successful MSC therapy is in the manufacturing. Here I just show you one slide where we compared term or preterm mesenchymal stromal cells, because I got this question in the morning. And so here we got preterm MSCs from a cohort of a 24-weeker without preeclampsia, without chorioamnionitis. And you can see dramatic improvement in survival with MSCs from term but also preterm animals compared to untreated animals. Improved lung function, lung compliance, improved lung architecture in term and preterm treated animals compared to untreated mice. And then I have to show you this picture. So here we also show that there's improved improvement in pulmonary hypertension, but I have to show you this picture of the lung vasculature. Again here, the dramatic pruning in our O2 induced lungs. I know at least of one preterm, ex-preterm that is now 35 years old in the States that emails me every month asking me when can he have my cells. And he has exactly this happening in his lungs. He was never intubated, but he had CPAP and probably high levels of oxygen in his, he has pulmonary hypertension and his CT scan looks emphysematous-like. And you can see here preservation of the lung vasculature with either term or preterm MSCs. And then you can quantify this where you see here the vessel diameters. So the small vessel, the distal capillaries are dramatically reduced here, but preserved with mesenchymal stromal cells. In conclusion, I hope I could convince you that mesenchymal stromal cells are a very promising multimodal approach for preventing BPD. However, it's a heterogeneous population of cells. They act through a paracrine effect, and while the time is ripe to perform carefully designed early phase clinical trials, we still need to learn more about the biology of these cells. And here is an important piece of wisdom that's suggesting that we should not end our understanding of MSCs now, otherwise bad things may happen. If you want to know more about the Star Wars version of mesenchymal stromal cells, you have to come to Würzburg. And just want to thank all the people behind that work because it takes a lot of work. I also have Marissa Lithopoulos, who is obviously Greek. She does the fantastic work on neuroprogenitor cells. She's really amazing. Maria Piero did the work on the first work was quarter MSCs. Then we have very young people that did work on the exosomes and conditioned media. And here, 1, 2, 3. Over the next 6 months, they may give us some cues about what is a good MSC and what is not a so good MSC. I'm very excited about these data, and I hope that within 6 months we will be able to answer the question. This is breakthrough number 1. Breakthrough number 2 and 3 over the next 10 years are in the making. And I would like to thank the people behind the methods approach. They are very crucial to make it as evidence-based as possible. And this is how the first phase 1 in Canada may look like. And this is the team behind it. And I hope that we will be able to start by the beginning of next year. Thank you very much for your attention.